Okay, so when we talk about exposure at the cellular and tissue levels, there's a whole host of different processes that we're concerned about. The first one is genetic changes. So when we look at the structure of DNA, there's different types of damage that occur to the sugar phosphate backbone of the DNA or the bases of the DNA. And the one we're most concerned about is called a double strand break, which is when you ionize both sides of the double strand. And that double strand break leads to cell killing, chromosome aberrations, mutation, and also plays a role in cancer development. So what's of importance for space radiation is that they not only can cause double strand breaks, but they can cause clustered double strand breaks. So X-rays and gamma rays or electrons on Earth can cause double strand breaks, but they're really isolated from each other. So you'll have a double strand break on one chromosome in a low dose exposure, and maybe one in another cell, or perhaps on another chromosome in the same cell. But with heavy ions, like iron nuclei in space, you can have a whole string of double strand breaks along one chromosome that occur. So this is quite different than types of radiation on Earth, and that's why you have a high quality factor and a high concern for the type of radiation in space. Now, the other types of things that can occur are more on the level of signal transduction, that when cells respond to DNA damage and oxidative stress caused by radiation, they send out biochemical signals. And they're really there to protect the tissue from the exposure. So what's concerned for NASA is that we're talking about a different type of insult from heavy ions that would be part of the galactic cosmic rays than what humans or other life forms on Earth are used to. So the signal transduction processes that occur with heavy ions from cosmic rays are quite different, we're finding, from X-rays and gamma rays. So we really don't know what the outcome of that would be, but it looks like there's different contributions from gene expression. If you look at the 50,000 or so genes that make up the human genome, you'll see different genes expressed after heavy ion exposure compared to X-ray exposure or unexposed cells. Also, the combination of different proteins that are expressed is different with heavy ions. So these types of changes will lead to tissue effects. So that translation from genetic damage to tissue effects and ultimately to a safe and healthy tissue, or a tissue that's going to form a tumor, is really on the balance between the genetic damage and these signal transduction pathways that alter the homeostasis of the tissue.